Good morning, everybody. Right. Can you hear me? Good morning, I'm Andrew Giles, and I'm the CEO of the Garvan Research Foundation. Welcome to our second seminar for 2013, Cancer in the Age of Personalised Medicine. As you would be aware, it is Garvan's 50th anniversary this year, and so we've had a whole range of events in order to promote 50 years of outstanding medical research in all of our sections. And really, we need to pay tribute to the vision of the nuns, the Sisters of Charity, who set up Garvan 50 years ago. The nuns, as you may recall, set up the Garvan as part of an amazing capital campaign that they wanted to run in order to build a medical research institute to mark the fact that the nuns have been in Australia for 100 years. They were very, very lucky in the late 1950s to receive one huge donation of £100,000. And that was from a young lady who wanted the money given in memory of her father, who was James Patrick Garvan. And I tell you that story because many people in this room are donors and supporters of Garvan. It's nice to think that Garvan was born out of the generosity of one or two quite passionate individuals, and to, even now, in our 50th anniversary, we're very reliant on the generosity that we get from the broader community. So I do thank all of you who are donors for your great and ongoing support. And I also understand that there are quite a few people in the room who have left legacy to Garvan in their wills, and that is a particularly unique gift, and I do thank you very much for that. Our entire immunology program, which is actually a world-class program, was set up on the basis of one major donation that we received from a requester. So the power of leaving a gift in your will for Garvan is very, very important. So I thank those donors who are in the room. A range of speakers today, as you can see. There'll be a morning tea just before 11 o'clock, and then we'll finish, if we have time, with some questions and answers. Cancer is one of those things that touches so many people, so it's great to see so many people here today to learn a bit more. There wasn't a lot of good news in the budget that came out on Tuesday night, but hidden away down the very, very bottom, there was a small increase in funding, $5.5 million for the establishment of a new prostate cancer research centre here at Garvan Kinghorn. So I think it's wonderful to see that even with the budget the shape it is, there is still an ongoing commitment from the federal government to support the world-class research that is going on here at Garvan. Let me introduce you our first speaker, who is Dr. Alex Swarbrick. Dr. Swarbrick is head of the Tumour Progression Group at the Garvan, based within the Kinghorn Cancer Centre, and he is a conjoint senior lecturer at the Faculty of Medicine at UNSW. Dr. Swarbrick.
Thank you very much, Andrew, and it's wonderful to be able to connect to you, our engaged community of people who are really excited by the research and the clinical developments that we've got happening here on campus. So, as Andrew mentioned, I'm in the cancer division here at the Garvan Institute. I actually work in the fantastic new Kinghorn Cancer Centre, as you can see here on the slide. This is a joint venture between Garvan and a hospital, and the idea is to make the research into cancer and the delivery of cancer care a seamless transition. And so, in fact, in this building, the first few floors house all the outpatient cancer services for St. Vincent's Hospital, and the floors above are cancer research. And I think, as you'll learn today, the boundaries between research and cancer care are really blurring, which I think is a really big win for all of us, for the community. Now, this is a building that was completed very recently. It was supported in large part by funds from the federal government. And despite the gripes about the lack of support in the recent budget, I'm really edified to see that even in an election year, they're still coming in and lending a hand on the weekend to get some of our samples. Okay, so let's back up for a sec and just talk about what is cancer, make sure we're all on the same page here. Cancer is a genetic disease, and you're going to hear a lot about genetics and genomics today, because this is one of the major features that's driving this revolution in personalised medicine. Cancer develops when normal cells of your body acquire damage to their genes, so mutations to the genome, and it leads to this gradual change in behaviour from being a team player to being essentially a an unruly mob or a riot is a way you could think of a cancer. The cancer spreads, here we go, I've got a laser pointer here. The cancer spreads from normal tissues. It ultimately invades through various barriers meant to keep tissues apart, and it heads into your circulation, your blood, and it travels to distant sites. And this is this process of metastasis, where cancers spread from, say, your breast cancer, and they spread to your bone or your lung or your brain. And this is the major challenge in how we treat and research cancer, is this spread of cancer to metastasis. Because normally in solid cancers like this skin cancer, for example, the surgeon and the radiologist can cure you of that primary disease. But it's this disseminated disease throughout your other organs that are the real problem. So what is personalised medicine, and why has it become this revolution that we're really part of now? Well, there are a lot of different interpretations of personalised medicine, but these are some of the fundamental tenets, I think, at least from my perspective, that underpin personalised medicine. The first is a recognition that when we think about your risk of developing cancer, that we are all individuals. Just like it's obvious based on our appearance, our culture and so on, it's obvious how different we are. Medicine has now understood how different we are in terms of our risk of developing disease. And so that's both a factor in terms of how we live our lives, so our lifestyle, our exposure to the environment, whether we smoke, whether we live in a place with high levels of UV radiation from the sun, for example, and also the genetics that we inherit from our family history. Both of them play into our risk of developing cancer. And personalised medicine is changing the way in which we can help people understand their risk and then mitigate that risk. And I'll show you an example of that later on. Once cancer arises, of course, the second fundamental part of personalising cancer medicine is realising that cancers are also different. Cancers are individual and unique. And this is going to be a real thread that you'll hear a lot about today. Going from this idea of saying, oh, someone has breast cancer or brain cancer, we realise that every cancer has its own unique characteristics and traits, and that is at once a challenge, but also an opportunity for us to find the unique weaknesses of each and every individual cancer and target that. And so it takes us to this idea that instead of there being a magic bullet, a cure for cancer, there are in fact going to be many, many cures for cancer, just depending on which type of cancer you have. The final thing that underpins personalised medicine, and it's a little bit dry, so it will only be covered in part today, but it's this idea that medicine is becoming data-driven. We now have the capacity to collect huge amounts of detailed information about you and your disease, and to now use that information to really effectively and accurately track the course of your disease and to treat it most effectively. Okay, and so I run a research lab. I study mainly breast cancer, and what we do is try and understand the genetics, the genes that go wrong in breast cancer. And there are really three things we can use that information for. The first, as I mentioned, is to help us identify risk. And a perfect example of this, of course, is the BRCA1 gene. Many of you will have heard about this gene. There's been recent news, of course. Angelina Jolie announced to the world that she underwent a prophylactic mastectomy because she discovered she carried a BRCA1 mutation. She knew that that mutation comes with an extremely high risk of developing breast and ovarian cancer. So based on that genetic information, she then went and took the forward stance and underwent a mastectomy, which now massively reduces her risk of developing breast cancer. The second is allowing us to classify and stratify disease. So as I mentioned, there's not one type of breast cancer, there's not one type of brain cancer. And we're using genetic information to classify diseases into different types. The final thing, of course, is that the more types of cancer that we find, the more therapies we've got to find for each of those types of cancer. And so this is something you hear a lot about today, is developing new innovative therapies for each of these cancer types. And so this is the idea here. This is the conventional model for how cancer patients are being treated. They will present, if they have a cancer in their colon, they'll go to a specialist in colon cancer. The clinical trials generally treat all these patients the same, and so they tell you, a patient with colon cancer receives this drug. And we know that a proportion of patients receiving that drug will have a good outcome, and they will have a major benefit to their life. A number of them will get no benefit at all, and that's often the majority of patients receiving these all-comer kind of drugs. And there's also a class of patients who actually end up worse. They have severe toxicities, for example, from their chemotherapy. And I think many of us will know people who've been through chemotherapy treatment and have had a very tough time of it. So the new model is instead, when a patient presents, they undergo genetic testing to identify what we call biomarkers. So these are tests that tell you a lot about a cancer and predict the way a cancer will behave. Based on those tests, patients are then split into groups. This group receives this treatment, whether it be a drug or a surgical procedure. This group of patients this, and this group of patients a different treatment. And the net result is that they all end up with a greater benefit than if they were treated as a homogeneous group. So I think there's a bit of a misunderstanding in the community that sometimes personalised medicine means that every single patient will be treated in a completely unique way, and I'm not sure we'll ever end up at that point. But the idea is that we can at least identify unique groups within the cancer types that we previously thought of as a whole. So I want to show you now what is one of the real poster children for personalised medicine in cancer, and this is a drug known as Gleevec. It was developed to treat a cancer known as chronic myeloid leukemia, CML, so this is a blood cancer. This cancer almost, or very frequently, has this particular genetic damage known as a translocation in the genome. What that does is it creates an unnatural protein known as BCR-ABL. This protein is what we call an oncogene. It drives the development of cancer. And in these patients, when you look at their blood spread out on a slide, you see all these abnormal cells that are the cancer cells filling up their blood. This is a very aggressive disease. Once Gleevec was developed, it's essentially a very specific inhibitor of this BCR-ABL protein, almost like a key fitting into a lock. Gleevec only targets patients with this mutant protein. When you treat someone with CML with Gleevec, you can see that when you look at their cells on a slide, you don't need to be a pathologist to see that these are different, and there's this very rapid response. But I think what's really exciting is that Gleevec has been in development or in use long enough that we actually now have long-term data. Again, getting back to this idea of using data to understand treatment, that Gleevec leads to astonishing improvements in patient survival with CML. So this is what we call a survival curve. It essentially tracks the survival of patients with CML over time. So these are months of time along here. First, if you look at the light blue curve, these are patients who received the conventional treatment, the chemotherapy that they would have received 20 years ago. You can see that out here, for example, at 60 months, 5 years, more than half of these patients have died of their CML. In contrast, the patients who received Gleevec, sorry, my pointer's failing here, but you can see that these patients, more than 90% of them are still alive 5 years later. So I think this is really one of the success stories in personalised medicine that I think we're trying to now expand to other cancer types. And so it's this idea that instead of basing treatment on where a cancer emerges, breast cancer, lung cancer, instead we study the biology of these cancer cells in detail to understand the key molecules that make that cancer tick. And what these discoveries have led us to realize is that often those key hubs, as you might call them, can be found in a variety of different cancer types. So for example, a colorectal cancer at the level of the genome might actually look like a liver cancer and a brain cancer. And what this development has led us to realize is that drugs like Gleevec, originally developed to treat CML, can now be used to treat other cancer types in completely different organs that are also driven by those same molecular problems. And in fact, Gleevec is now being used, for example, to treat gastrointestinal tumours, and it's being very effective in a proportion of those patients as well. Another example, of course, would be Herceptin, a breast cancer drug, is now being given to pancreatic cancer patients in clinical trials. No one would have ever guessed that might be possible 10 years ago. And so I work primarily in breast cancer, so I'll summarize very briefly some of the movement in the breast cancer area. breast cancer was in fact one of the places where personalised medicine was first delivered. I think most of you will have heard of tamoxifen. It's an incredibly effective agent used to treat a subtype of breast cancer known as ER-positive breast cancer, and it's led to really phenomenal outcomes in those patients. And in fact, patients with responsive tumours, tamoxifen now gives them often more than 95% chance of living 10 years out from diagnosis. Another example would be Herceptin, which I just mentioned, another targeted therapy for a subtype of breast cancer. Now, in the last year or so, a lot of research has studied the genomics of breast cancer, and what has become clear is that there's a particularly difficult type of breast cancer known as basal breast cancer, for which we really have no modern treatments available. The challenge here is that it affects young women, and so often patients with basal breast cancer can be up to 10 years younger than other breast cancer patients. It's has a very mixed behaviour. Pathologists can't predict how these cancers are going to behave. Some patients will die 6 months after diagnosis. Some patients will be happy and healthy 6 years after diagnosis. And there are no approved therapies. But I think one of the really exciting discoveries in this area is that, again, now studying the genome, the DNA of these cancers, have made us realise they actually look a lot like type of aggressive ovarian cancer. And those ovarian cancers are now undergoing clinical trials with a range of new drugs. For example, these PARP inhibitors and platinum chemotherapy. So the idea is that maybe these basal breast cancers, again, have the same key molecular hubs as some of these ovarian cancers, and may respond to these same drugs. Okay, so I'll skip over one of my slides here just to finish up and say, well, what do we need to do to really deliver personalised medicine? It's a massive change in the way we study cancer and the way in which we deliver cancer care. And I think we all need to be engaged in this because it goes beyond the lab and it goes beyond the chemotherapy suite. It's something society needs to come to terms with and think about how we're going to do it. The first is that it's a data-driven enterprise, and so we really need to make sure that, that all the data that's now being generated on individuals and on the cancers needs to be accessible, it needs to be shared, it needs to be properly interrogated by computer scientists, essentially, to find the meaning within it. And certainly here on the St. Vincent's campus, we're developing some really state-of-the-art approaches to allow us to put together all that incredibly important data and find the meaning within it. The second is that we need to change the way we do clinical trials. Of course, in the past, it was easy to do a clinical trial in breast cancer. You just enroll every breast cancer patient you have and give them the same drug. Now, of course, if you've got a drug that works in 5% of breast cancers, it becomes a lot more difficult to put together a huge group of patients to test a new drug in. And so we need to change the way we think about doing clinical trials. The other thing we need to do is for cancer patients to participate in those trials, to want to be part of those studies. Because often these new agents have nothing like the side effects of chemotherapy, but instead they have often a great potential to lead to improvements in outcome and advances in our understanding of what makes the disease tick. Really one of the key developments in this area, just as we're doing in Kinghorn, is this tight integration of cancer researchers and clinicians. That's why we built the Kinghorn Cancer Centre, so that we actually bump into each other on the stairs. Patients, clinicians, and researchers are all using the same lifts and the same flights of stairs, and I think that's a really important development. And finally, I think it's a lot about education, to teach people about the promises and the challenges of personalised medicine, to take the advice, to be able to understand the information they're giving, to mitigate their own risks of developing cancer, and to then actually to follow the recommendations that they're given based on that information. And so I'll finish up there, and I think we'll probably have questions later on in the Q&A session. So now I'll hand it over to the next speaker, Sandra O'Toole. Thank you.
Thank you, Dr. Swarbrick. Our next speaker is indeed Professor Sandra O'Toole. Clinical Associate Professor Sandra O'Toole is a tissue and molecular pathologist who divides her time between Royal Prince Alfred as a staff specialist and the Kinghorn Cancer Centre, where she's co-head of breast cancer research. She is the founder and head of a new cutting-edge diagnostic service which offers comprehensive mutation profiling to guide cancer treatment.
Thanks very much, and I think Alex has given a wonderful introduction to this whole area and conveying the excitement that we see I'm talking mostly about lung cancer, but there's some really exciting developments in melanoma that I want to share with you very briefly as well. They say that a picture tells 1,000 words, and certainly when it comes to this new revolution in cancer treatment, there's no doubt that these amazing images show us the sort of changes that we can hopefully see in the future in more patients with cancer. These are images taken of patients with metastatic melanoma, and you don't need to be a specialist to realise that the images on the left here and over here show that these patients have very significant metastatic disease and probably not very long to live. These patients underwent genetic testing of their tumour and found they had a mutation in a gene called BRAF called V600E. These patients received a very exciting new drug called vemurafenib. There's a whole pipeline of agents like this coming out. And within only 2 weeks, you can see it, images on the right-hand side, virtually shut down the metabolic activity of these malignant melanomas. Similarly, in lung cancer, this therapy has been around a little bit longer than melanoma therapy. Again, don't need to be a specialist to see this is a CT scan taken through the chest of a patient with lung cancer. You can see that over here there's a mass that's also going near the windpipe, where it branches into two. And this doesn't look very good for this patient at all. This patient had genetic testing of their tumour and were given a mutation in a growth factor gene called EGFR, and they were given a drug called gefitinib that's actually available to people on the PBS at the moment. Only 5 days later, you can see that the tumour is melting away, hardly anything left and hardly anything there. I have to say though that unfortunately in most of these cases, the tumours are really smart and they learn how to get around this block. And in a matter of months, often the tumour is back but resistant to the new treatment. However, just occasionally there are big wins. This is a patient, this is a CT scan again through their chest. This is a picture of their heart. And again, you don't need to be a doctor to see that there is a big, large mass there in the heart. Surely this patient had a very little amount of time to live with a nasty tumour in that critical location. That was January 2010. This patient had an even rarer mutation in the BRAF gene called V600R and received the drug vemurafenib. And here they are almost 2 years later with no evidence of disease. Now I have to emphasise, this is a real standout example. Most patients, unfortunately, with metastatic disease who receive these therapies do recur in a matter of months. But this shows that we can find the right changes. Hopefully in the long term we can have a real impact on outcome. So we've entered the era of personalised or precision medicine. And this is a revolution in the diagnosis and treatment of patients with a range of cancers. And as Alex was describing before, patient selection and treatment for these new therapies is very much driven by genetic biomarkers in their tumour. So not in their inherited genes, but in the tumour cells itself. There's this explosion of research that means that there's huge pipelines of all these new treatments coming out. But the big problem at the moment is those study responses are only available to a specific range of malignancies and only subsets of those. So great effects, but we need to be able to do it for more patients. And the sort of cancers where this approach works are cancers that have what's known as a driver mutation. And you can see here in this image that you've got someone pushing the car. If the person's not pushing the car is not going anywhere. And this is a process called cancer gene or oncogene addiction. And while we know that cancer cells have multiple, hundreds of thousands of genetic abnormalities, in these specific examples, their growth and survival can actually be impaired by inactivation of one single cancer gene. And these cancer genes are really important treatment targets that lead to the sort of responses I was showing you in those images initially. And we're actually getting better at moving these changes into the clinic. Alex gave the example of chronic myeloid leukemia. This change is one of the first major structural changes seen in chromosomes in cancers, and that was recognised, ages ago, in the 1960s. But it took 41 years before there was a treatment that was actually able to help patients, the first reports in the literature. EGFR, which I was talking about is very important in lung cancer, we knew about that in 1978, but it took 36 years to get into the clinic. But I also wish I look at this side of the graph here. You can see that virtually 100% of patients have this BCR-ABL change. In lung cancer, it's probably about 10 to 15%. Alex also mentioned GIST, so these stromal sarcomas that were very hard to treat but show amazing responses to Gleevec. But as we go down the list, you see that although we're getting quicker and quicker at getting into the clinic, in fact, it's smaller and smaller portions of patients with these malignancies. The most stunning example is one of these other structural changes in the chromosome of cancer in a gene called ALK. It was described in lung cancer in 2007, and 3 years later, the first clinical trial was published in a top journal, and patients are receiving drug now. So that's an amazingly short period of time from identification of change through to change in practice. So we are getting better, but it's getting harder because we've already got the common ones. We're looking now for these really rare things in smaller and smaller subsets of the tumour. So lung cancer is a great example of where personalised medicine is working well, but also illustrates the challenges. Lung cancer, we've known for a long time, it's the leading cause of death in men in our society, but unfortunately, it's now overtaken breast cancer as the leading cause of death in women. Even Van Gogh knew that smoking wasn't good for you, and we know that 90 to 95% of all lung cancer is tobacco related. In New South Wales alone, in 2011, there were 2,600 deaths. And the big problem is the number of patients diagnosed and the number of patients who die each year are very similar because it's such an aggressive disease and it's diagnosed late. So that 70% of patients, when they're diagnosed, will not be suitable for any sort of curative treatment. As a pathologist, my job is looking down the microscope to help try and divide cancers up, to give clues about how we should treat them and what the outcome is likely to be. And when we're talking about lung cancer, the commonest type of lung cancer is called non-small cell lung cancer. This is small cell lung cancer, which is still very little progress in this area and very poor prognosis, a very aggressive disease. But this type of cancer that's quite common, one of the commonest types of non-small cell lung cancer called adenocarcinoma, is where all these exciting new therapies are becoming available. Obviously, we need to work in the other areas as well. So when I'm talking about lung cancer today, I'm actually talking about this very specific subset of patients that have interesting changes. Now, while I look under the microscope and I can tell that cancer looks different, they've done a lot of work now, huge international efforts to understand the genetic profile of a whole range of cancers, including lung cancer. And what's become apparent in this pie chart is that if we look at lots and lots of different genes, in fact, more than half of patients with this common type of lung cancer, gland cancer or adenocarcinoma, have a mutation with one of these drivers. And while some are very hard to target, like KRAS, others like EGFR or ALK are showing really amazing responses. But you can see that the slices of the pie are getting smaller and smaller as we're finding these new changes. So it's getting harder and harder to find new things. What else is intriguing is that the genetic profile of a tumour is different depending on whether you're a smoker or not. If you're a non-smoker and you're unlucky enough to get lung cancer, one good thing is you're far more likely to have one of these changes that can be treated with EGFR or ALK. Smokers often have a mutation in KRAS, which at the moment is very hard to target. There's also an intriguing ethnic difference in this type of response and the mutations. Younger patients, women, and patients of Asian background are far more likely to have the treatable changes compared to the rest of the population. So we've seen not just changes in the tumour, but important ethnic differences that can help predict who may be responding to these treatments. So in non-small cell lung cancer, about 10 to 15% of patients have mutations in EGFR that can be treated with 2 drugs that are available to the general public, just bempegaldesleukin and lotinib. The good thing about these, they're taken orally, so you don't have to go and have chemotherapy needles. They have relatively few side effects, not free of side effects, but much less than general chemotherapy. And they can show, at least initially, amazing response rates. Up to 70% of patients will see some dramatic improvement, like some of those scans that we saw. There's a new change that I mentioned in the gene called ALK and a new one called ROS that also seem to be very sensitive to a new drug. And as I mentioned before, there's this pipeline of new agents that are coming out all the time, so it's a very exciting area. And that has implications for testing, because at the moment, the way that the government funds testing and the way that it's done in most labs in Australia is that we only want to know about EGFR because there's a drug we can use for that. But as you can see, there's a lot of research going on, and the pace is coming up so fast that we want to know about more than just the one gene. So if you have a look at this pie chart here, wouldn't it be great if you have your cancer sample tested to know if you've got any of these changes that might possibly help you with this terrible disease? And this really poses questions for testing. This is my job at Philanthropy Hospital, where we do testing to try and find these changes to help patients receive the right treatment. As I mentioned in lung cancer, 70% of patients can't be operated on at diagnosis. That means you get the tiniest little piece of tissue from a very fine needle pass through, often under guidance with a CT scan. The results have to come out quickly because lung cancer has a poor prognosis and patients don't necessarily have long to go. It has to be accurate and sensitive. And as you can see from those pie charts, wouldn't you like to know about everything that could possibly be treated? So what we do at Philanthropy is we've got a technology that's got a very long name that is basically just called a sequenom. And the way it works is that we do a few chemical reactions on the sample. You know, with a mix of tumour DNA, normal tumour DNA, the normal cells, and then the cancer cells that have the mutation. And these will have a different, their fragments will have a different weight. So we put this out into a little vacuum tube where we've got normal and wild type. And a laser beam fires, shoots these fragments down the vacuum. And because they're different weights, they'll travel at different sizes. And we can pick up these changes as little blips on a graph. And it's actually, we're using a commercial panel called the Oncology, like the cancer map panel. And this lets us, on one sample, look at 238 mutations in 19 genes, that at the moment covers 90 to 95% of all treatable mutations. In the same amount of time, we'll take other labs to do one test on one gene that we know is not that common anyway. So the output is really easy to read. I'm sure I can convince you of that. This is a normal sample where you've got one peak, you've got one type of DNA normal. Here, we've got two peaks. We've got normal DNA and we've got the mutant DNA. And this mutation is one of the commonest mutations in lung cancer that is sensitive to these new treatments. So we've been doing some patients at Prince Alfred Hospital and Concord Hospital for the last probably about 18 months now. And we did a research study on patients with early lung cancer to have a look at their patterns of change. And we found, in fact, that more than half of the patients have a clinically important or potentially targetable mutation. And this paper's been really well received and is hopefully going to influence developing clinical trials for patients with early lung cancer before the disease is spread, where I think we have a chance of cure. Because once the horse is bolted, we just try and give patients more time and good quality time. Very exciting new changes coming along too. I mentioned these structural changes in the chromosomes. One of these is a newly described one, ROS1. These pictures are from a journal published just last year. This was a 31-year-old non-smoking patient who presented near death, unable to breathe from extensive infiltration from lung cancer. They were lucky with a cutting-edge centre in the US where they just described this change. They tested the patient, they had the change, they gave them the drug, and 3 months later the lung fields are cleared. Now, chances are this will recur, but giving the patient this quality time, this amazing response, and the hope for the future, I think is really important. This is where Alex was talking about the link between the lab and the clinic. Because I work at the Garvan, where we saw the basic scientific discoveries and we hear about all the changes really soon, we've got the skills to put them into practice as quickly as possible. This paper came out in 2012. 6 months later, at RPA, we were able to offer the test to patients in a proper accredited setting. And this was a 39-year-old woman who presented totally unexpectedly with cancer of the lung. We did our multi-gene test that was negative. We did another common test called ALK that was negative. And because we had this new test, we were able to find out that she was positive for this. And this young woman is receiving drug and at least initially showing a good response. Very briefly, amazing things are happening in melanoma. As you saw from these images before, these tumours shut down within 2 weeks. There was a major trial published in mid-2011. Australia has a very high rate of melanoma, and a combination of pale skin and sun is not a good one for the incidence of melanoma. And while most melanomas, when they're cut out, patients will be fine, if they develop metastasis, the outcome is really quite poor. Alex talked about survival curves here, and this is this amazing new drug, vemurafenib, for patients with specific mutations. You can see that initially, if you've got the terrible prognosis disease of metastatic melanoma, at least initially you have this really good response compared to standard chemotherapy. But unfortunately, as I mentioned, often the cancer gets smart and works out ways to get around that. Where we hope to make real changes is by looking back at patients before they have spread, where they've got high risk of spread but it hasn't spread, in what's called the adjuvant setting. That's what happens in breast cancer. Women give chemotherapy up front to try and change the course of their disease. And that's where we see important improvements in outcome. Melanoma, we now know, is not just BRAF. We also care about other genes like NRAS and KIT and GNAQ. And there's a whole pipeline of drugs that are coming through that help treat these patients. And there are a number of clinical trials. We work very closely with the Garvan Institute of Australia, and we're able to give them information on patient samples to help funnel them into the right clinical trials to help as soon as possible. And these examples of 300 patients with melanoma we tested last year. If you just bring one gene, you find half the changes. We're able to offer a whole suite, and these patients are offered therapies that might help them. The real challenge is going to the future is that we've plucked the low-hanging fruit. We've got very common mutations. We know about those now. And the big problem is trying to find the smaller and smaller pieces of the pie of patients who might benefit. And this can be very difficult. So I mentioned that where this approach works well is in tumors with one single major driver mutation where they've got this addiction. But unfortunately, most cancers don't look like that. Most cancers have what are called thousands of passenger mutations because the whole genome is going crazy. They don't necessarily drive the cancer, but they're all there. How do you find which ones are important? Or that they've got multiple drivers and knocking off one is not going to make a difference. And that's going to lead very much into Mark's talk, because I think we've been offering these panels at the moment, but I think the way things are going is in this amazing new technology called next generation sequencing, where you can look at thousands of genes and thousands of mutations at the same time. But the real challenge is working out which of those are important. So we've definitely entered this revolution in cancer medicine. We're at the beginning of it. I think where we're going to see real improvements is when we can move these to cancers before they're spread. It's a hope it's clear that we need to be looking at more than just one gene, which is what the government's funding at the moment. So we make sure the right patient has the right test to get them the right drug at the right time. Thanks very much.
Thank you, Dr. O'Toole. That's great. Our next speaker is Dr. Mark Cowley. Mark is a cancer genome scientist here at Garvan, working in the King Cancer Centre. He studies the genetic makeup of patient tumours using next-generation sequencing. His research involves characterising the mutations and other aberrations present in patient tumours.
Thank you, Andrew, and I say what a pleasure it is to be able to talk to you today about this exciting research. Alright, so today I'd like to talk about genome sequencing and what this actually means for cancer patients, and it's really nicely on Sandra's talk. So while I was writing this, my wife was watching Grey's Anatomy. And let me just share a little bit about what Grey's Anatomy has to say on the topic.
So the chemo didn't work? Just like the radiation.
Andrew's cancer has continued to spread.
So what? Surgery now? I mean, you said that surgery could kill him. You said he was too weak.
Well, Dr. Bailey here is suggesting another approach. It's called genome mapping. If we can identify the specific DNA sequence of Andrew's tumor, it can open up a variety of treatment options and target drug therapies.
And no surgery?
So I think this is actually no surprise given what you've already heard this morning, but the promise of being able to analyse a tumour, identify the specific molecular mutations, and then try and treat with the right drug at the right time is the promise. And if it's on Grey's Anatomy, I think it's high time you all found out a little more about it. So, stepping back a little bit, this is all about DNA, genes, chromosomes, and genomes. So as I'm sure most of you know, DNA is the molecule that stores information in all our cells. It describes where the genes are, when these things should be turned on, in what order. And you can see that at the fundamental level, genes are just 4 little bases, A, C, G, and T, organised into very specific orders which control the instructions in the cell. And as you've already seen this morning, mutations, even a single mutation to one of these bases can be catastrophic. In the case of the BRCA1 mutation, it's had a lot of attention this week. So there's 2 major landmarks which really brought us here today. The first for me would be the Human Genome Project. This was an 11-year publicly funded effort. It cost nearly $3 billion and took 11 years, but the goal was to sequence a healthy human genome, to give us the reference sequence so that we knew what a healthy genome looked like. Given that information, the goal was to work out what the genes are and then we'd be able to work out what mutations affect which genes and help improve treatment of cancer and other diseases. Interestingly, it found that only 2% of that genome actually encodes proteins, or the things that we thought were the functional units in the cell. 98% of the genome, once considered junk, now a lot of research has shown that there's some really interesting components of the genome which aren't proteins, but which could potentially be important in disease. But that's probably for another talk. The second major revolution is next generation sequencing. The first study of this type came out in 2005. There's a lot of technical detail behind next generation sequencing, but what I want you to take away from today is that essentially what we'll do is we chop DNA up into tiny little fragments, maybe 100 nucleotides or 100 bases, 200, 600 bases. We now know how to mobilize these things onto glass slides or into little droplets, and we can perform these sequencing reactions in a massively parallel way. So instead of taking 11 years to sequence a human genome, now we can do it in 10 days. Absolutely huge revolution in molecular biology. This is the most common machine right now. It's probably about the size of this lectern, so not huge. It's called the Illumina IC 2500. Hopefully you'll hear more about those in the press. But as I said, they fragment the DNA into short little tags of about 100 bases and then sequence each of those tags to work out the order of A's, C's, G's, and T's. You've probably heard in the media that we're approaching the $1,000 genome. This seems to be the case. We're not quite there yet, but hopefully we will be there within the next couple of years. The other flip side of that is the $1,000 genome and the $10,000 analysis. We've still got quite the capabilities to analyze all the information. But we're certainly working in that area at the moment. My role in this is as a genome data analyst. These types of things generate about 6 terabases of data on a single run. To give you a feel, that's about 3 million digital movies. There's a huge amount of data. We then have to process all that data, we have to refine it and work out which are the specific mutations important for the disease. I'm a big data guy. I love data. I love analyzing, generating, and looking, and I hope that I can share some of that data with you today. We are looking here at a little slice, 200 bases in the human genome on chromosome 12. So the genome is laid on its side. And these are these short little tags that I described, about 100 base pairs long. And through the analyses that we do, we try and work out exactly where those tags live in the genome. And this bright red band here indicates that there's a specific mutation. This one affects the KRAS gene, which Sandra's already talked about. It's one of the most common mutations in pancreatic cancer, and sadly it's one of the ones we can't target yet. But now we know how important it is. A huge amount of effort is undergoing in KRAS. I suspect we'll be able to cure it at some point, but just not yet. Importantly, when we sequence the same patient's healthy genome, or their blood or their skin, that mutation is not present. So that tells us that this was a mutation that was acquired in this specific tumor, and it's one of these driving mutations. It's one of the hallmarks of this person's cancer. The second data slide, hold on for a second. I want to actually just describe to you what a cancer genome looks like. One of the ways that we view them is in these circular plots, where chromosome 1 is here, 2, 3, all the way around to chromosomes X and Y. We can then start to layer in the types of mutations that we can detect in cancer. The first you've seen quite a bit today. These are these single nucleotide changes that affect one base in the genome. They can be catastrophic in the case of that BRCA1 mutation that Angelina had. So these are characterized by these little lines. They affect very small parts of the genome, but they can have quite a substantial change. Next, cancers are, as Sandra pointed out, they're quite cunning. So another way that they can affect large numbers of genes is to delete chunks of DNA. By doing so, you might delete 20 odd genes, 100 genes, and those genes are usually responsible for controlling the cancer cell. If you delete those genes, then all of a sudden it can start growing out of control. Additionally, you can take accelerators of cancer and amplify those regions of the genome. So instead of having one version of the gene, you might have 3 or 5 or 10 copies, and that's going to act as an accelerator. That's what's shown in this track here, where here's a complicated region, but Down here you can see an amplification. In here you can see a deletion of a bunch of regions. So there's a little complicated, but it's just trying to show that we can also detect different types of mutations. And the final part, we've already heard about this BCR-able oncogene this morning, and that arises from translocations where one bit of this chromosome, well, these two bits of the chromosomes actually swap. One bit gets translocated to another chromosome, and that creates a mutant gene, which is then uncontrollable. And that's what these lines indicate, where there's a connection between chromosome 1 and chromosome 13. So that's really what a cancer genome looks like. And if we look at hundreds of these things, which I'm about to show you, they all look quite different, which is both an opportunity and a challenge. So hopefully the take-home message is that we can characterize tumours and we can look at different types of events which drive those tumours. I've been fortunate to be involved in a large study called the International Cancer Genome Consortium, and together with that and the American effort, which is the Cancer Genome Atlas, we've already analysed close to 10,000 patient tumours across a whole range of different cancer types. I was involved in a pancreatic study, and Dr. Pajic in the next session will tell us more about that specific study. But you can already see that 1,000 breast cancers have been analysed, 600 colorectals, 580 ovarian cancers. So we're actually learning a lot of things in the research setting from analysing these cancer genomes using technology that we never had before. So hopefully learn more about these tumours and then ultimately how we can treat them. This is the one slide I wanted to show you about what sequencing 100 cancer genomes might look like. So essentially we sequenced 100 pancreatic cancer genomes, and this picture describes the frequency of each mutation that we detected. So we found a KRAS mutation in nearly every patient. The second most common mutation was this P53, and then SMAD4 and P16. So these are the 4 big drivers of pancreatic cancer, but what you also see are all these little mutations here, which are the passenger mutations that Sandra talked about. These are present in a patient's tumour. They may represent therapeutic opportunities if we can target these things with a drug. But I think the take-home message from that is that each tumour is quite different, which again is a challenge and an opportunity. You've already heard quite a lot today about what this type of testing from Sandra's lab can do today. What I probably should have added in here was we can look at the whole genome, but we can also look at subsets of the genome on the fly. So we can look at the 2% that encode the proteins, we can look at 200 cancer genes, 100 cancer genes, 50 cancer genes. The flexibility of this technique is really why I think it's going to be useful in the clinic. We've already seen examples of the BCR-able and a targeted therapy that works in a specific cancer type. We were involved in showing that HER2-positive cancer type in pancreatic and potentially being able to treat that with Herceptin, which is exciting. And so today the druggable genome represents about 50 combinations of genes, mutations, and cancers, and I hope that through next generation sequencing, this druggable genome is actually going to grow substantially. What can we do in the future? By sequencing patient cancer genomes, we aim to identify things where we can repurpose drugs, and Alex already touched on this. It's the idea of using a drug that's been shown to work in one cancer in a different cancer, especially if the same mutation is found in both drugs, both cancers. Rescuing failed drugs is a big one. So there's tens of thousands, hundreds of thousands of clinical trials, many of them fail. Within each of those trials, there's often a few remarkable responders. Now we understand more about cancer and the heterogeneity. We think that if we were to work out which ones actually did respond, work out what was in their genomes that caused them to respond, then all of a sudden we can translate those findings to new patients. The pure research question is that by analysing these genomes, we're going to learn more about cancer, and that will lead to new targeted therapies in the future. And as Sandra showed, how quickly we can actually do that now that we have places like the Genome Centre connecting research with clinicians. And as I said before, by doing this research, I think we'll expand the druggable genome. So the final slide is that hopefully I've shown that next generation sequencing will be an enabler of personalised medicine. As opposed opposed to treating a whole cohort of patients with one drug, we can use next-generation sequencing to identify subgroups of patients with specific mutations that can then end up going on specific therapies. And as Alex pointed out, this is a data-driven exercise. So by tracking all of these patients and working out which didn't respond here, maybe which didn't respond here, we can actually refine these connections between mutations and drugs. Thank you very much.
That's great. Thank you, Mark. Before we break for morning teas, I'd like to introduce you to Carol. Just give you a brief overview of what the foundation is doing at the moment.
Thanks, Andrew. Good morning, everybody. It's great to see so many familiar faces here today. And as Andrew said, we've got some partners for the future here. Thank you very much for your wonderful support and many other different types of supporters. And we're very, very grateful for your help. We wouldn't be here if it wasn't for you. So the Garvan Research Foundation, of which I'm a part of, and Andrew's actually my boss. He's a great MC, isn't he, by the way? So we're the fundraising and marketing of the Institute. But you may not know this, but every dollar that we're very lucky to receive from NH and MRC funding, we have to raise another 70 cents from the community, from people like yourselves. And what doesn't get covered by the NH and MRC funding are things like novel projects. So if one of these brilliant scientists come up with an idea to go in a completely different direction that has no already funding in place for it, that through Philanthropy we're able to help them get the runs on the board and get the raw data so that after a couple of years they're able to then apply for funding through the government. So that's a very important function of your support. The other reason we need your support too is for equipment, because this government funding doesn't cover equipment, and as you can see, equipment is definitely the way of the future with the next generation sequencing and so forth. So this is where you come in. At the Garvan, we like to think that everybody has a choice in the way that they help us. The whole philosophy of the Garvan is founded on choice, and the freedom and the ability to choose is what largely defines us as human beings. And these different choices that we make are very individual. So these are some of the ways that people are helping us here at the Garvan. I'm helping me with my microphone, but anyway. We have corporate partners, and today you're actually sitting in the NAB auditorium, and we have some lovely volunteers from the MLC, which is part of NAB, helping us. Just on that too, all the ladies who were helping with the reception and helping you through the doorways and so forth today are all volunteers here at Garvan. So that's another way the community is helping us. Word of mouth is extremely important to our work, so all you people here today are now our ambassadors. So off you go and spread the word about the incredible work that's taking place here in the Garvan, St Vincent's and the Kinghorn. Of course, many of our supporters are donating on a regular basis. They might be giving a certain amount, on a monthly basis, an automatic deduction and so forth, and that really makes a huge difference in the pool of funds that are available for our research. Community fundraising. You've probably seen a lot of community fundraising taking place for the Garvan over the last 2 years since Andrew started. Yeah, there you go. And in fact, we've had people running in the City to Serve. We've also got a wonderful young man called Samuel Johnson, who's now riding a unicycle around Australia to support his sister, Connie, who's only in her 30s and unfortunately is in her 3rd round of breast cancer and probably won't see the year out. So this lovely young man, who you may have seen on The Secret Life of Us in the past, not on Grey's Anatomy, unfortunately, but he is doing this huge trip to fund research into breast cancer. Don't think that's how I'll be helping the Garvan. But we have a number of people who are giving through donations in workplace giving, so that when you actually, use salary, it's taken out of your salary before you can see it, so it's an easy way of giving. And finally, leaving a gift in your will, as Andrew mentioned, the Garvan has an incredible legacy of bequests that we actually started up from a bequest. Our immunology department was started through a wonderful bequest. And it's a very, very special kind of gift. I know myself, soon after I joined the Garvan, I was so impressed with the work that was being done here that I decided I'd leave a small portion of my small estate to the Garvan. And I was saying to Andrew last week that we call it a selfless gift, but it's really not that selfless, because you feel so fortunate to be able to do something like that. And, I feel that I now have shown something to my son as well, that he has seen that I care about future generations and that I care enough to give part of my estate to the Garvan for their research. I also get my name up on the honour board as a partner for the future, so yeah, there are a few payoffs to it, and it's very, very important to our work. And every bequest, whether it's large or small, goes into an endowment fund, which becomes a corpus and grows, and we're able to use that interest for the future fundraising and needs of the Garvan. So if there's anything there that you might like to choose to help us, you may already be helping us in some of these ways, or if there's another way you might like to help, please come and see me at over tea and coffee, or just come and see me anyway, because I just love to have a chat. Okay, thank you.
All right, so let's have a quick break. So there's morning tea. There's morning tea down here, isn't it? Yeah, there's another from Marina down here or upstairs. So depending which way you'd like to go. Maybe the back row may want to go up to level 2, and the front row may want to come down here to level 1, and then we'll reconvene in 15 minutes. It will be exactly 11:15. Well, welcome back, everybody. Thank you very much. I hope you are enjoying the seminar and you're learning something from our amazing speakers. I have the great pleasure of working here every day, and every day I continue to be astounded by both the cleverness of the researchers here at Garvan. I'm not a scientist, I actually don't understand a word they're saying, but they're very well respected. They're getting some good government funding and they publish a lot, so they must be doing something right. Well, what always amazes me is the level of passion they have for the areas they are researching. So 2 more speakers, and then we'll open up to questions and answers. Our next speaker is Dr. Marina Pajic, who is a member of our pancreatic cancer group here in Garvan Institute. Her research centres on the development and analysis of mouse and cell-based models of pancreatic cancer. These models are then utilised in the development of new targeted therapies for pancreatic cancer. Dr. Pajic, welcome. Welcome.
Hello everyone. Thank you very much for having me here today. I'm very grateful to be able to tell you a little bit about some of the work that's been happening in the Pancreatic Cancer Research Group at the Garvan Institute, and and also our efforts as part of the Australian Pancreatic Cancer Genome Initiative. So the reason for this target board here is really because what we're trying to achieve is to apply more personalized treatment approaches to how we treat pancreatic cancer. So to really be able to target and tailor specific therapies, give the right drug for the right patient, hopefully at the right price. So pancreatic cancer is a disease with dismal prognosis. So it is the fourth cause of cancer death in Western societies with very poor overall survival rates. So only 5% of patients are alive 5 years post-diagnosis. The average survival is only about 6 months, and 90% of the patients die within a year. Mortality has unfortunately remained unchanged for almost 50 years. And surgery is currently the only potential for cure. And so this is for only a small proportion of patients who do not, at the time of diagnosis, present with metastatic disease, which is actually the majority of pancreatic cancer spectrum. Chemotherapy is also given, and it's modestly effective. But what we have seen from our clinical research is that there are some significant responses that are observed in specific yet really undefined subgroups of patients. So we would like to figure out what is going on there, try to identify these survivors, try to identify what's defining these responses, and then apply that for the new patients who come into the clinic. So this is the poster child, so the International Cancer Genome Consortium, of which we are members of. So this is a large international collaboration. The aims and the goals of the ICGC are to obtain a comprehensive description of all the genomic changes. So these are, for example, mutations in the different genes in 50 different tumor types. So these are all of, we're looking at mutations that are of clinical and societal importance across the globe. And what this slide shows you is just the various countries and multiple research teams that are contributing to this effort. And you can see also Australian involvement here as part of this project. Now the next slide shows you the Australian Pancreatic Cancer Genome Initiative and the teams that are involved in this project. And so we as members of the APGI represent the pancreatic cancer sequencing component of the ICGC. And on this slide you can see the multiple different centers that are contributing their time, their patient samples, as well as clinical data to our joint effort. So you can see that there's involvement of multidisciplinary teams all across Australia. Again, just to reiterate, this is a global, national and global collaborative effort, and we have clinical collaborators, currently 130 that are part of our team, a scientific team of 60 very motivated people. We have generous support from the online community, and this also includes our collaborators as part of the International Cancer Genome Consortium. This is 200 and growing, and we are gaining increased exposure on various social networks as well. So this includes things like Facebook, for example. So what are the aims of the APGI? So our aims are really to catalog all the genetic changes that occur in pancreatic cancer. We want to use this information to return it to patients and ultimately really change the way we treat this disease. We're also looking at identifying new mutations and potential targets that we can then develop and test new therapies for. So what this pictorial here shows you is what each patient who enters the APGI study goes through. So you can see the patient gets diagnosed, subsequent treatment plan is developed, patient undergoes surgery, and there is extensive clinical follow-up. In parallel, so on the horizontal line here is what we call the genome journey. So you can see what happens to the sample as it leaves the patient and undergoes extensive quality control on our end. We then prep the analytes, which includes the DNA from these tumor samples. They get sent off to our friends and collaborators at the University of Queensland. So this is the lab of Professor Sean Grimmett, who does all the sequencing analyses. Subsequently, where we can, we really want to use this information directly to guide clinical management for these patients. So you can see that there. Unfortunately, for those more novel mutations, we can't return the information back to the developed new preclinical models in the laboratory, and we then use these models to study these mutations, determine the function, and then hopefully when the next patient enters the study, we're able to guide therapy for these patients. So what is our current progress at the moment? So we have 597 prospectively recruited patients. So this is quite an extensive pancreatic cancer cohort. Over 6,000 different analytes have been prepped for various molecular analyses that are going on right now. 355 of these patients of the 597 recruited are currently undergoing sequencing. So these are the patients we're looking at are pancreatic ductal adenocarcinoma patients. This is the most aggressive disease. So this is what we're really working on first. We're also acquiring additional cohorts that are driving further research. So these include metastatic patient samples, which really are proving to be an enormous resource for us in the laboratory. 208 tumor and normal DNA sequences have been now complete, and 108 of these are already publicly available. So the main goal of the ICGC is really that this data does not remain locked away somewhere in a cupboard and that nobody ever uses it and sees it, but to actually make it available to everyone, every single scientist in the world, so that they can answer key questions of interest to them, because this is really the only way that we can progress research forward. And we're also generating preclinical models in the lab, like I've mentioned before, and so this includes the 20 patient-derived cell lines that we have generated for the sort of functional studies. So this picture right here just shows you, it's a graphical representation of our recent data that's been published in Nature that Mark Cowley has mentioned before. So this is the sequencing effort of the first 100 pancreatic patient tumor exons. And what this illustrates really, and so each of these panels is a different tumor type. So pancreatic cancer is not a single disease. It's a very heterogeneous, molecularly complex disease, and almost unique from a genetic standpoint. So there are multiple potential phenotypes or tumor subtypes, and each of these is either at cusp or below threshold of detection in standard clinical trials. So these are these exceptional responders that we seem to be able to occasionally see in pancreatic cancer. So currently, this one drug for all patients approach, which is the standard approach in the clinic for pancreatic cancer, simply does not work. So we need a more personalized or tailored therapy-based approach. So these are some of the, you don't really need to pay attention to all the different drugs that are listed here, potential phenotypes, but what we're working with, so some of them are already used in the clinic. So we know, for example, that some patients respond really well to gemcitabine, which is the standard of care for pancreatic cancer. There are also some of the other interesting drugs, things like olaparib, that are used in breast cancer. So we're looking at repurposing agents that are used for other cancers, specifically for specific subtypes of pancreatic cancer. Then we're also looking at some of the novel treatments, and so these we're really studying in the preclinical models with the hope that they will guide future trials. So now I'll just very briefly run you through our sort of research pipeline and how we're trying to translate discoveries into direct patient care. So this is tumor DNA, and as you would have seen earlier from Dr. Cowley's talk, you now all know what this circular plot means. And I have questions about it. So what you see here, right, is lots and lots of these different lines. What they show us is that this tumor has many, many different types of mutations in it. So we call these tumors with extensive genetic abnormalities, or unstable tumors, unstable DNA tumors. Now, in comparison, you can see here a tumor DNA that is almost completely stable. So there are not many of these lines, not many of these mutations, and so you can clearly start to see a difference when you perform whole genome sequencing on these tumor samples. So I'll focus on this highly genetically unstable tumor. Because one characteristic of these tumors in some of the evidence from other cancer types, for example, breast cancer, suggests that these tumors are very aggressive, very fast proliferating, so very fast growing. But one interesting characteristic is that they preferentially should be very sensitive to specific therapies, specific types of DNA damaging drugs, and one of these is mitomycin C. So we wanted to obviously test the hypothesis. Firstly, we have to test it in preclinical models. And how we do that, we have developed various models in the laboratory, including these patient-derived cell lines. We run these things called drug screens. So we are able to get the tumor out of the patient and then grow it in these dishes with special kinds of probe nutrients. We can then add different drugs on top of it, and we can then look at what drugs are the best at killing specific types of cells. And so this is one specific result where we're directly comparing tumor with this unstable DNA compared to with a tumor with a stable sort of DNA fingerprint. And we've added two different types of drugs. We added gemcitabine, which is the standard of care, so we test everything against gemcitabine. And shown in red here, we have mitomycin C. So what very quickly becomes obvious is that this particular type of tumor preferentially responds to mitomycin C. So this itself is not enough to persuade oncologists, but we have obviously studied this much, much further. So collectively, with this data and additional molecular data, we were able to apply this directly to the patient's treatment. So here we have a patient with this same genetic fingerprint, so highly genomically unstable tumor, who has relapsed on adjuvant gemcitabine following also surgery. So this little circle here shows you a recurrence on a CT scan. Based on our information, the oncologist was satisfied and administered more targeted therapy. So in this case, this was mitomycin C, and the patient has had a complete response. So a complete resolution of the lesion on the CT imaging. We have also measured serum markers from these patients. This patient, so we're following him closely. But this patient is doing really well. So our ultimate goal is really how we can start to apply a more personalized treatment approach to how we treat pancreatic cancer. And really, the current challenge for us now is how we can apply this for every patient that enters the clinic. And so that is the challenge. So I would just like to very briefly thank the whole, well, this is actually not the whole team, but these are some of the members that I am directly working with, members of the Garvan Institute Pancreatic Cancer Research Group, clinical collaborators around Australia, our friends in Queensland, our funding bodies, and most of all, really, I would like to thank this generous support of the patients, because without patients, we would not be able to do any of these studies. Also, the family members and friends and members of the community. Thank you.
Great, thank you very much. Our final speaker is Professor Richard Epstein. Professor Epstein is Director of Research and Senior Consultant at the Department of Oncology at St Vincent's Hospital. He's Conjoint Professor at UNSW, and he is Visiting Scientist, Garvan Institute of Medical Research, and of course he is the head of the Laboratory of Genome Evolution and Informatics in the Kinghorn Cancer Centre. Please welcome Professor Epstein.
Well, good morning. It's great to see you all here. Thank you so much for your interest and your support. Like some of the other speakers, I work in the building next door, and if any of you haven't been in there, you should walk in and have a look at the lobby. It's designed for the particular architecture which is designed to facilitate interaction between doctors and scientists. And it really is a very exciting place to work, and exactly the type of work that we need to be doing with cancer at the moment. Now, I'm going to try to make my talk as simple and as brief as possible. The one thing I want to emphasize is that we are just at the very beginning of a really exciting era in research and in clinical medicine. I think that in the future it's going to be just a little bit more fun to be a cancer patient than it is at the moment. But we're not there yet, and there are some important problems that we still need to address very urgently. I'll talk about these in my talk. So where do we begin with personalised anti-cancer interventions? I know you've heard some of this before, but obviously the place to begin is with prevention. It's much better to prevent a cancer than it is to do battle with a cancer which is very far advanced. And in the future, you and I will be able, I'm sure, to have our genomes sequenced, and we will have a piece of paper that tells us you have a predisposition to diseases A, B, and C, and you need to do X, Y, and Z to prevent or detect this early. But we're not there yet. Where we are at the moment with prevention and personalised anti-cancer interventions is still at the level of the family history. If you or I have a strong family history of cancer, that's a clue that perhaps we have a gene that may predispose us to one or other types of cancer. And a couple of these are quite common, and you probably read in the paper on Tuesday about the actress Angelina Jolie, who found that she was a BRCA1 gene mutation carrier, and that her mother had a battle with breast cancer. So Miss Jolie underwent a prophylactic intervention, and that this is a complicated subject, but she felt that testing was very worthwhile for her. Apart from breast and ovarian cancer, the commonest cancer with a family history is bowel cancer. So these tests are routinely available in our genetic counselling clinics for people with a strong family history in any type of cancer. One has to think carefully before having a gene test, and this is why we do genetic counselling. The advantage is, if I'm in a strong family history of cancer, and I do a gene test that shows me that I have the defective gene, which has been identified in my family, then I know that I would be well advised to either have early detection by screening or some sort of prophylactic intervention. Similarly, if I'm already known to have a defective gene, the question arises, do I want my children or my brothers and sisters to be tested? Partly because half of them may be found to have entirely normal genes, and they may have a weight lifted from their shoulders and not need to worry about being an additional risk. So there's another type of personalised gene testing that we can do, which has already been spoken about, which is that if I have cancer but I don't have a family history, I can ask my oncologist to test my tumour. So we can not test my blood, but my tumour, and we can see, as has just been described for pancreatic cancer, what is the pattern of genetic aberrations or vulnerabilities in my cancer. There are two common types of tests. I know Sandra spoke about these tests before. The type that we use in routine practice are these few single gene tests that we can do on the cancer to see whether there's a mutation or an increased number of genes in a tumour. And the great advantage of these particular tests is that they strongly predict the sensitivity to one or other of these very expensive but very good new anti-cancer personalised treatments that we have. So these tests are not too expensive. They're just about standard, and we recommend routinely that people have these tests. Some of them are paid for by the government and some are not. The other type of cancer gene testing which is done, and which you could have also heard about this morning, is when we look at multiple genes, anywhere between 20 and several hundred genes, pattern of expression of these genes in your cancer or my cancer. This is a fantastic thing to look at if you're a researcher. We love to look at these and try to understand how a particular cancer is driving itself. And it also gives us a very strong impression of whether my cancer is a pussycat or a tiger. It tells us about the aggression of the cancer and the prognosis. Where I think these tests, which are some of them are much more expensive, where they're not quite up to speed yet, is they don't necessarily predict the benefit of individual treatments or expensive treatments in the way that the single gene tests do. So I'm sure this will change in the near future, but at the moment I don't personally routinely recommend the purchase of these very expensive tests for all my patients. But that will come. In fact, in the United States, this is already a huge industry. There's already almost $10 billion a year being spent in the United States on cancer gene diagnostics. So it's coming. It's a very popular idea, even if not all of the evidence is yet there. Well, moving on from personalized gene testing onto personalized treatments. And before I tell you why we're so keen about personalized treatments, I want to say a little bit about unpersonalized treatments, or the old-fashioned treatments, because I want to try to illustrate what the advantage of the newer treatments is. So the old treatments, as you know, include local treatments, surgery and radiation therapy. Now these are undoubtedly the best treatments to have for your cancer, because they imply that perhaps you just have a local disease and it may well be cured. So these are the most curative treatments you can possibly have, and I definitely recommend having them if you have early disease. If you have widespread disease, metastatic disease, then these local treatments are not useful because they can't be given to multiple sites in the body. And so then we have to rely on drug treatments, because that's what drug treatments do. They go everywhere in the body. Metastatic disease is difficult not only because it's in more than one place, but by virtue of the fact that these cells are growing in parts of the body where they're not meant to grow, we know that these cells are all defective and dying, so they're not as sensitive to our treatments as we like. They're also constantly selecting for new genetic forms because they're going to different parts of the body. So it's like truly moving goalposts. You might have a response, but then the response goes away. And using these old-fashioned drugs that we call chemotherapy or cytotoxic chemotherapy, which means cell-killing chemotherapy, we have to get over these disadvantages. And the first disadvantage of using cell-killing drugs is side effects, because they kill all the cells in the body. So we have the usual unpopular side effects of chemotherapy that we all know about. But I'll tell you at the end of the talk, the side effects are not completely bad, and I actually hope that my patients have a little bit of side effects generally, so long as it's tolerable. The second problem we have with these cell-killing drugs relates to resistance. Because what these drugs do is they select very quickly. That they kill all the cells that are easy to kill, but they leave behind the most aggressive and resistant variant cells. And here is a scan of a patient on the right-hand side of the screen. You can see with disseminated lung cancer. He has 4 months of chemotherapy with all of the usual side effects and hair loss. He's shown his X-ray in the centre there. After 4 months, all the cancer's gone. Everybody's very happy. Then 4 months later, the disease is back. And it's back. It's even more aggressive and resistant than ever. And his life expectancy at this time is much shorter than it was at the beginning, even though the X-ray looks much the same. So very difficult to prove a net improvement in survival and quality of life for old-fashioned chemotherapy for most common disseminated cancers. So that raises the question, well, ideally, what type of drug would we like to treat advanced cancer? Well, we'd like one, first of all, that's highly specific for the cancer. Doesn't affect our other cells, because that way we have very good quality of life. We live better. That's one of the key aims of treatment. The other thing I'd like in an ideal drug is a drug that doesn't kill the cells that are easy to kill, because that's just selecting for resistance. I'd like a drug that switches cancer cells off, or makes them differentiate, or sends them to sleep. And so we're not getting a selection for resistance, because that way I think I've got a better chance of prolonging my patient's survival, and hence the patients will live longer and live better. That's what we'd like to see. And that's exactly where we're going with these new drugs, which you've been hearing about today. We've got drugs that just act like a switch, much easier than killing the cells, and they have names which we started to hear about in the late 1990s, like Vincent and Gleevec. In fact, these are still some of the best personalised cancer drugs that we have. But we do still have a few problems to overcome. And I've already alluded to one of them, which is selection for resistance. It's not only a problem with the old-fashioned cytotoxic drugs, and I think you heard from Dr. O'Toole this morning about a new drug called vemurafenib or zelboraf, which is a wonder drug in melanoma. And this slide just shows again that you can have a tumour, which is circled there on the left-hand side. It gets much smaller after just 2 months treatment. The patient may feel better. But then after 6 months treatment, it's back again. This is a real problem with this drug, that it tends to only work on average for 6 to 8 months, and then the disease comes back. So it's been a little bit difficult to prove a major survival benefit, even though the response looks fantastic. And in fact, the Australian Pharmaceutical Benefits recently did not approve this drug for general use yet. We're hoping it will in the future, but I can understand why they didn't. It's an expensive new drug, and the effect on survival is not yet as dramatic as we'd like. The second problem we can have, even if you have a target-specific drug, sometimes the side effects can still be dangerous, even if they're not as predictable as with old-fashioned chemotherapy. You can have inflammation of the lungs or heart, which can be potentially life-threatening in a small percentage of cases. And some of these drugs can even pop holes in your bowel. Again, very uncommon, but we need to warn people about these problems because sometimes you're unlucky. The third problem is we would like, because all of these new drugs tend to be rather expensive, we'd like to be able to give you a guarantee that yes, it's very likely to be worth the outlay, either whether we're talking to the health funds or to the individual. This is easy to predict for some drugs, but not for others. And the problem we get into in the clinic is this leads to a number of very long conversations. From the government's point of view, there's only two types of drugs. There's those that are proven to work and those that are not proven to work. And quite reasonably, the government says, well, we'll pay for those ones that are proven to work, but nothing else. But in real life, of these ones that are not proven to work, only some are proven not to work. And some we don't yet know whether they work or not, or we know that they work a bit, but we don't know how much. So we spend a lot of time in our clinics discussing with patients, look, there's a drug here, it's not yet paid for, we think it's pretty good. If cost isn't an issue, you can think of having this treatment. But it's very hard without the data, without the research, to really quantify and give people an accurate idea of whether some of these drugs are going to work for them. That, of course, will all change, but we're not there yet. The final point I want to make is that, unfortunately, treating a cancer is a little bit more complicated than treating an infection. We'd like to think in the age of personalised treatment that, as with an infection, I could say, when you've got an infection, it's resistant to penicillin, but it's sensitive to something else, sulfonamide. So I give the sulfonamide and the problem is fixed. Why isn't it that simple with cancer if we have this personalised approach? I think one of the problems is that you can't have a cancer of the bacteria. For cancer, you need to have an entire organism with multiple cells, because what the cancer actually does is it exploits the cells, the normal cells in the body that are around it. So these cells that are around the cancer are actually part of the problem. But there's also an opportunity, because we can target those cells as well as the cancer. And I want to talk a little bit about that. The first way we target the cells outside the tumour is the old-fashioned way. We manipulate the hormones. We know that the most successful anti-cancer drug of all time, arguably, was this drug, tamoxifen, which is used for breast cancer. It's used for prevention. It's used for palliation. Very well tolerated. We still don't have a better drug than this. So that's the type of new drugs we'd like to get. The second type of regulation of the tumour from the outside is, as I was saying before, the cells that are immediately around the cancer, the primary tumour or metastasis. These cells are involved in growth-promoting loops with the cancer. And if I have preventive chemotherapy for bowel cancer or breast cancer, I know that I actually need to have a certain amount of toxicity, a certain amount of side effects to get any benefit. Probably the reason is that these normal cells around the tumour are dying off because of the chemotherapy, and that may secondarily be preventing the cancer cell survival in those metastatic niches that we're trying to target. So side effects, like we see with chemotherapy on the left-hand side, particularly the fall in the blood count is something we like to see a little bit of. Or even when we use targeted treatments, some of which we know have to cause a rash, otherwise we don't get any benefit. These tell us that there may be some effect on the tumour from the normal cells. This is why when I see my patients, I sometimes express moderate pleasure that they have certain side effects. The third thing that controls the tumour is the immune system. Of course, we've been looking at the immune system for many decades, but it's become a particularly hot topic of recent years. And I'll just give you one example, which you may read about in the newspapers, which is that here's a drug, it doesn't matter what it's called, but this is a drug that activates the immune system. And so here's a patient with on the left-hand side, there's a large metastasis from melanoma in the liver, which shrinks down with this treatment. And a very gratifying thing about this type of treatment is it seems to work across a whole range of different cancers. And the other thing about it is, even though the shrinkage of the tumour might not be as dramatic as with some of our other treatments, these patients seem to live significantly longer. So the survival benefits look to be even more impressive. So I think this is really going to become something we're going to hear much more about over this next few years. The final thing, and this really is the final thing that I'm going to talk about, is the metabolic system. As you probably know, a lot of cancers are rising in frequency. These are what we call the lifestyle cancers. And these cancers include cancer like breast cancer, colorectal cancer, prostate cancer, endometrial cancer, and quite a few others. Probably the reason that these cancers are going up in incidence is that our lifestyle is different to what it used to be thousands of years ago when we were swimming through the trees. We're much better nourished, we're under much less physical stress. What this means is that the normal cells in our body are not dying off as quickly as they used to. And that's what cancer likes. So any precancerous cells are nourished by our relatively comfortable lifestyle and our lack of really vigorous exercise. And we actually know that caloric restriction, dieting, maintaining our ideal body weight, and regular vigorous exercise not only reduces the incidence of these lifestyle cancers, but even after I'm diagnosed with one of these cancers, my outlook is better if I subsequently maintain my ideal weight and increase my exercise. And what's really exciting is this is not just some kind of punishment. We actually started to understand the molecular pathways that regulate these benefits. For example, we know that if I'm a few kgs overweight, as I probably am, I know that this, what we call dysfunctional fat in my body, is playing through certain growth factor pathways related to insulin that stimulate cells to survive and stimulate cancer cells to grow, and also that it triggers some low-grade inflammation, which also gives rise to cancers. So by dieting, I can actually switch off these pathways if I have the willpower to do so, and reduce my cancer risk in the future. Similarly with exercise, we know that vigorous exercise will activate certain molecular pathways that cause cells to die off. So we know these are just like taking pills that block these pathways, but we can do it through our own efforts. And these are a really important part of modern cancer management. So I'm going to finish it on that note. I just want to reiterate that we are just at the very beginning of a really exciting era. We do have a few problems still to overcome. The experience of having cancer, if we're treated with these targeted drugs, tends to be significantly better than with the old-fashioned treatments like cytotoxic chemotherapy. And perhaps in 20 years' time, we may not have any cytotoxic chemotherapy units like we do today. But the survival gains, we still have a way to go. It's still, we need to improve the durability of the benefits from these drugs. The second problem we're going to have, and we're going to hear no end talk about, is the cost of these drugs, how they should be regulated, which patients with which biomarkers or which genomic mutations should have subsidized treatment, and which ones we should have to pay for ourselves. There'll be a lot of discussion about this. And finally, clearly we need more research. There's so many things we need to understand better. We need to find new targets. We need to work out how to use different personalised drugs together without undue risk, but with better benefit and more durable benefit. We need to find how to use these drugs for prevention instead of just using them at the end of life. Much better to prevent cancer than just palliative. We need to do more clinical trials, because without clinical trials and the evidence of benefit from clinical trials, none of these drugs will ever be licensed. And of course, we need to look at the cost and benefit. So thank you very much.
That was great. So I'm very conscious of time, and I know that some people do have to leave on time. So rather than open the floor to questions, all of our speakers, apart from Dr. O'Toole, are going to stay down the front. So if you have a particular question, once we wrap up, maybe like to come down and ask the questions. That way we can free people up and head out the door. I'd ask you to join me in thanking all the presenters for being here today. According to my running notes, it now says I present them all with a bottle of wine. It is indeed a very cruel world. The relevant person will be shot. The wine will be delivered to your offices, which are in the building. We are redoing our entire seminar program. So on your seats there are some feedback forms. If you have the time to fill them out and hand them in to my staff and our volunteers on your way up, that would be great. But we are just looking for some ways that we can tweak and change these events for the rest of the year, and also more importantly for the years ahead. Thank you all for coming in, and thank you for your interest in Garvan. Again, if you have a question, please come down the front. Otherwise, thank you for being here and travel safely home.